Welcome everyone, and thank you for being here today. We're about to take a detailed look at something you interact with every single day, but may not fully appreciate: the journey of food through the human digestive system. From the first bite to the moment nutrients enter your cells, digestion is a seamless, complex, and fascinating orchestration of organs, enzymes, and biochemistry. For NEET aspirants, digestion is not just a chapter, it's a foundation. This topic combines physiology, chemistry, and even pathology. So, whether you're aiming for clarity or accuracy, understanding this journey in depth will serve you well, both in your exam and in your understanding of the human body. Let's start with a philosophical but essential idea. You are not what you eat, you are what you absorb. Digestion is the bridge between food and life. It allows large, complex molecules like starch, proteins, and triglycerides to be broken down into smaller, absorbable units. These nutrients then fuel your metabolism, support growth, and repair tissues. Without digestion, food would have no biological meaning. From a clinical perspective, poor digestion can mean malnutrition, even if you eat well. From the NEET perspective, this system is rich with factual and conceptual questions, so it pays to understand both the process and the purpose. Keep this in mind as we journey through each stage of digestion, not just where it happens, but why it matters. The journey begins right in the mouth, a site often underestimated. But here, both mechanical and chemical digestion begin in earnest. Mechanical digestion is facilitated by teeth, incisors for cutting, canines for tearing, premolars and molars for grinding. This physical breakdown is essential because it increases the surface area of food particles for enzyme action. Now, let's talk enzymes. Salivary amylase, secreted by the parotid glands, initiates the digestion of starch into maltose. It operates best at a pH of 6.8, which is mildly acidic to neutral. That's important because once it reaches the stomach, amylase is inactivated. The buccal cavity also hosts mucus, which lubricates food and forms it into a bolus for easier swallowing. NEET tip: be clear on which enzyme begins carbohydrate digestion, and remember, salivary amylase does not digest disaccharides or proteins. Those come later. As food enters the stomach through the lower esophageal sphincter, it's met with an acidic, enzyme-rich environment designed to break proteins into smaller chains. The stomach secretes hydrochloric acid, HCl, which plays multiple roles. It kills pathogens, denatures proteins, and activates the proenzyme pepsinogen into pepsin, the primary protein-digesting enzyme in the stomach. Pepsin breaks proteins into shorter polypeptides. For infants, another enzyme called renin or chymosin is present. This helps coagulate milk proteins, allowing for more efficient digestion and absorption. Mechanically, the stomach churns the food, mixing it with gastric juices to form a semi-liquid substance called chyme. NEET tip: pepsin works optimally at a pH of 1.5 to 2.5, unlike most other digestive enzymes. This acidic environment is unique and often tested. Also, pepsin cannot digest food without HCl. It's a classic activation question. The small intestine is the real star of the digestive system. It's where 90% of digestion and absorption occurs. It has three parts: duodenum, jejunum, and ileum. The duodenum receives secretions from the liver, bile, and pancreas, enzymes, and bicarbonate. Bile emulsifies fats, breaking large globules into smaller ones, enhancing enzyme access. The pancreas releases enzymes like amylase, trypsin, and lipase. Amylase continues starch breakdown, trypsin breaks down peptides into smaller units, and lipase breaks down triglycerides. The intestinal wall itself secretes enzymes such as maltase, lactase, and peptidases. The jejunum and ileum are heavily adapted for absorption. Their walls have villi and microvilli, increasing surface area exponentially. Each villus has a capillary for amino acids and glucose, and a lacteal for fatty acids and glycerol. NEET tip: know the function of each enzyme and its origin. Questions often ask, which enzyme comes from where? Also, be sure you can distinguish absorption pathways of fats versus carbohydrates and proteins. By the time food reaches the large intestine, most nutrients are already absorbed. But its role is far from redundant. The large intestine's primary job is reabsorption of water and electrolytes. Without this function, we would constantly be dehydrated. It also helps form semi-solid feces, which are stored in the rectum until elimination. Another key role, often overlooked, is the action of gut flora. These bacteria synthesize important vitamins, including vitamin K and some B vitamins, especially B12. Neat tip: although absorption here is minimal, the large intestine's role in water balance and vitamin production is a frequent MCQ topic. Also, understand the distinction between roles of small versus large intestine. Once nutrients are absorbed, they still need to reach the cells. This final step is called assimilation. Glucose travels through blood and enters cells via insulin-mediated transport, where it's used in cellular respiration. Amino acids are taken up by cells and used to build new proteins, enzymes, receptors, structural proteins, and more. Fats, absorbed into lacteals, travel via lymphatic vessels, eventually joining blood circulation through the thoracic duct. They are used in energy storage, cell membrane synthesis, and hormone production. Neat tip: don't confuse absorption with assimilation. Absorption is into blood/lymph. Assimilation is into cells. Also, remember the role of insulin in glucose uptake, frequently tested in integrated biology questions. Let's now explore what happens when this delicate system goes wrong. Ulcers are open sores in the stomach or duodenal lining, often caused by Helicobacter pylori or long-term NSAID use. These disrupt the mucosal layer that protects the stomach lining from acid. Diarrhea is usually caused by infection or irritation, leading to rapid movement of food and poor water absorption. It can quickly lead to dehydration, especially dangerous in children. Constipation, on the other hand, results from slow movement of feces, leading to excessive water reabsorption. The usual culprits are low-fiber diets and inadequate fluid intake. Let's look at a structured table now to organize these facts for NEET. Focus on causal chains, cause-symptom-affected organ. These flowchart-style connections are often used in assertion reason and case-based NEET questions. This slide is your NEET cheat sheet. Let's review high-yield facts. Enzyme source and pH sensitivity are critical. For example, enterokinase, secreted by the small intestine, activates trypsinogen to trypsin. That's an MCQ magnet. Bile, though essential, contains no enzymes. It emulsifies fat. Most digestive enzymes work at a neutral to slightly alkaline pH, except pepsin, which works best in acidic conditions. Don't forget the brush border enzymes like maltase, lactase, and sucrase, often ignored but highly testable. Neat tip: create flashcards or a mini table with enzyme, source, substrate, product, pH. It helps reinforce retention, especially under timed conditions. Let's bring it all together. The journey of food involves 5 key steps. Ingestion, the act of eating. Digestion, both mechanical and chemical. Absorption, nutrients entering blood and lymph. Assimilation, nutrients entering cells. Digestion, removal of undigested waste. Each organ plays a unique role. Remember the flow: mouth, stomach, small intestine, large intestine. Enzymes act like molecular scissors, cutting large food molecules into usable forms. The system is elegant, efficient, and essential. Make sure you see the big picture. It will help you make sense of even the trickiest NEET questions.